Hello, and welcome to the Field Guides. I'm Steve, and I'm here with Bill. Good morning, Bill.
Good morning, Steve.
And Chris. Good morning, Chris.
Good morning.
So what we're going to do today, and over the course of many future episodes, is give you the experience of what it's like to be out in the field, in the woods, and on the trail. For every episode, we pick a natural history topic, research the science on that topic, and take you out to a natural area where we share with you everything that we learned. And today, we are at Rollin T. Grant Wilderness Park in Lockport, New York. Now, I couldn't find much information about this place. Do you guys know anything about it?
I think wilderness should be in quotes.
Well, we have a wilderness creek, and we have a wilderness road.
Yeah. Now, full disclosure, folks, this is take 2.
Oh, yeah.
This is one of the few episodes we've had to completely re-record, and I will take full credit. There apparently was a setting on the mic that I messed up on. So thank you, Chris, for being willing to come here and repeat everything that you told us like 2 months ago.
I was going to say, it's a long time coming because the last time we recorded was the end of November.
2025.
2025.
Yeah. Now it's March 2. Oh, wow. So it's more than a couple months.
Happy to be back.
Yeah, right.
But I did look in the parking lot this morning because we actually recorded at a different spot nearby last time. But this park, it's about 175 acres. It is owned by the town of Lockport. So, folks, we're in a little different area of Western New York than we normally record in. We're actually north of the city. So, what would you say, about half an hour north of Buffalo?
Yeah, roughly.
Roughly.
Yeah.
So, a little closer to Lake Ontario than Lake Erie than we normally are. But this particular park that we're in, it's about 175 acres, owned by the town of Lockport, and we're standing along the edge of a ravine. There's a creek at the bottom. This is 18 Mile Creek.
Oh, really?
Not the 18 Mile Creek that we recorded the
Now you
Sturgeon episode at.
Now you got me thinking we're in Hamburg, all right? Because
No.
That's the 18 Mile Creek I know.
So one little tidbit I thought was interesting that I read just a few minutes ago was a trail that Seneca natives used when they were traveling.
Oh, cool.
So, yeah.
And little known fact about 18 Mile Creek, it's named that because it's 18 miles from any other creek.
No.
What?
We did learn last time, though, right?
We did, yeah.
Isn't it 18 miles from where the Niagara River empties into Lake Ontario?
I'm actually pretty sure that, or is it like the head of the I don't know. I can't remember. I was hoping you would know, so I didn't just give like a lie to the audience.
It's something to do with the mouth of a river.
Right. Yeah.
We'll cut this out.
Sure. All right. Well, without any further ado, I want to say who Chris is. Just some guy named Chris we met on the trail. But this is Dr. Christopher Osborne. Actually, do you want to introduce yourself? Actually, so
Sure.
Chris and I used to work together, but we actually worked together back in November when we recorded, but we don't work together anymore.
It went so bad last time.
Yeah.
Chris stopped working with Steve.
That's my interpretation of it too.
That's correct. So Steve and I, when we were working together, we were working at the University of Buffalo, where I was finishing up my PhD, and Steve was doing a postdoc there. Since finishing my PhD, I have started a position with it's called a CSU position, which is a position funded by the federal government that sends money to universities to hire people, and CSU stands for Cooperative Ecosystem Science Unit. And I'm an aquatic biologist doing fisheries research for the Lake Erie Fisheries Research Unit for the New York State Department of Environmental Conservation. And my primary focus is conservation genomics. I use cutting-edge genomic techniques to help inform conservation and management decisions, particularly in fishes.
All right. Well, congrats on getting your PhD.
Thank you.
So last
And
time,
getting hired for something afterwards.
right away.
Yeah.
So last time, were you officially a doctor or no?
Yes.
Oh, okay.
Mm-hmm.
Yeah.
He had already defended. He was just wrapping up with us.
Gotcha. That's got to feel good.
Yeah.
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 Mm-hmm.
You're like a real boy now.
First person in my family to graduate high school, so getting a PhD is a
All right.
Hell
step
yeah.
up, I think.
In all seriousness, congratulations on
Thank you,
that.
sir.
And thank you
Appreciate
for agreeing to be on the podcast, but also being willing to do this all again.
Absolutely,
Yeah,
yeah.
yeah.
I've been. I said this last time, but you guys are going to have to suffer through it again. I'm a big fan of the podcast. I've listened for a long time, and Steve and I's friendship actually grew from this podcast because we met in the first time we were in graduate school at Buffalo State. And then I went off to Peace Corps and served in the Gambia and listened to this podcast the whole time I was there. So it's weird that Steve and I were friends for about a year. And then during the time I was in Peace Corps, I felt like I got much closer to Steve and knew Bill really well. And then I came back and, we actually crossed paths again and did become closer friends. So, yeah, it's an honor to be on the podcast. I'm excited.
Yeah, Chris and I have been in 2 graduate programs together. Back when he was just a wrestler from Ohio, and then afterwards when he was like a super well-established scientist, one of the most gifted that I've worked with. So,
wow, that's very nice, Steve. I feel the same way about you, obviously.
Well,
you don't.
I know you have to say
you're a great wrestler. I mean,
yeah, right. Hey, don't. No one needs to know we wrestle. All right, so I'll start by saying that when we get into the realm of genomics, sometimes things can get a little complicated. So I feel like anytime we come across a complicated topic, 1, Bill, I want you to slow us down. But 2, there's just going to be certain things we have to go over. And I'll give sort of a short list of things that you'll probably hear later in the episode. Like, we need to talk about what an extinction vortex is. We need to talk about what a structural variant is, which is an important thing in genomics. We have to talk about bottleneck effects, also super important in genomics. And then a couple paradoxes that I know you guys have never heard before, like the genetic diversity population viability paradox and Lewontin's paradox. All of these things we'll talk about, but I just sort of want to put a pin in those now that these are things that we're going to have to sort of slow down and make sure that the audience is at least has a basic understanding of before we kind of move on with.
Good, because I'll warn you guys. I will be here to be like, all right, guys. You lost me.
Yeah.
Please do,
yes.
Yeah.
All right, so we are talking about the brown tree snake today.
Yes.
Yes, and Chris, would it be okay if I gave kind of a background on the brown tree snake, just as a species, and then you can jump in and tell me what I'm getting wrong?
You probably know as much as I do. I'll admit we had some very helpful collaborators from the USGS that work on Guam that know much more about the brown tree snake ecology and invasion. So yeah, please take it away.
All right, so we'll just start off by saying the brown tree snake, especially the brown tree snake on Guam, is basically the what-if nightmare case study for invasive predators, right?
Yeah.
And that's what we're kind of going to base our conversation on today. But the snake itself, what's the scientific name? Do you guys know?
Boiga irregularis.
All right.
Imagine if we didn't know.
It's a slender, nocturnal, arboreal snake.
All snakes are slender. All right.
Well, yeah.
Arboreal snake. Okay.
Maybe to you, Steve.
Sure.
So it's native to places like northern Australia, Papua New Guinea, and then parts of Indonesia, areas in the western Pacific. And the group that it belongs to, its genus, Boiga, often called the cat-eyed snakes. It's mildly venomous, which we'll talk about a little bit. It's one of those rear-fanged snakes, and shout out there to our Rattler episode, because we talked about the different fangs set up on different snakes. And it's really adapted for climbing and hunting in trees. They typically grow 1 to 2 meters long, so 3 to 6 feet. They have those large eyes, vertical pupils, and they feed on a wide variety of prey, including birds, lizards, bats, and small mammals, which they did a great job of eating on Guam. A little too good. And then they also, as we already mentioned, they're really good at climbing trees, but also entering buildings, which doubly creates problems, not just for the ecology on Guam, but for people. And we're going to even get into how snakes can cause power outages on Guam. Now, unlike many invasive species stories out there, as far as we can tell, it was not introduced to the island of Guam intentionally.
Correct.
Right? And I don't know about you guys, but I had to look up exactly where Guam was.
Oh.
I know it's out there in the Pacific somewhere, right?
Way out there.
But I actually did find out it's much closer to Japan than it is to Hawaii. So if you think of Hawaii kind of like in the middle of the North Pacific, kind of the lower part of the North Pacific, if you keep heading northwest, getting closer to Japan, Asia, then you'll get to Guam. But it's a good 3,000 to 4,000 miles away from Hawaii.
Yes.
So it's out there. But even though Hawaii is so far from Guam, why do you think people on Hawaii, especially ecologists, need to be worried about the brown tree snake getting there?
It's because there's frequently transport between Guam and Hawaii, and some of the insane negative impacts that we'll probably talk about in detail could happen on Hawaii that happened on Guam if the brown tree snake ever made it there. And there's actually a bunch of teams that are actively monitoring for a potential invasion front introduction event for the brown tree snake on Hawaii.
Mileage means really nothing, right?
Right. Yeah.
Right.
Exactly.
In terms of transport. So.
And Hawaii's already lousy with invasive species.
Oh, yeah.
So, yeah.
They don't need one more, right?
Right.
That's
So as far as people can tell, it probably hitchhiked accidentally on cargo transport post World War II, probably near the end of World War II.
Yeah, military cargo most likely, because there's a military base on Manus Island, which seems to be the source of the invasion or introduction. The source population, rather, aka where the population came from, and there's an Air Force base on Guam, so they probably came over on military cargo transport.
And then it exploded on Guam because, as we said, it was kind of this what if nightmare, because this island had no native snakes.
Correct.
So you had all these naive prey species that, in my head, I just picture the snake climbing up into a tree heading towards a bird, and the bird's just like, hey. What are you?
This is awesome.
Honestly, it's probably not that different than what actually happens. I mean, they don't have a fear response to snakes. They don't have certain defenses against snakes, and they were very vulnerable. They were naive for that reason.
Yeah. So you had no native snake predators. You had prey that were naive, and then you had warm climate and good habitat.
Yeah.
So just this perfect mix of ingredients. And then by the mid 1980s, the one number I found was, within 30 years, there were about 40 snakes per acre at the peak.
Wow.
Which is crazy, isn't it? And I don't know if it's true, but the one reference said that's one of the highest densities of snakes like anywhere in the world.
Yeah, I was going to say, what's the density of snakes per acre here? I feel like there's no way.
Well, they're the natives. So there's been some work that's been done to see how dense their population is in their native range, and it's like 2 snakes per hectare as opposed to 50 snakes per hectare on Guam. So it's insanely high.
Yeah, I even saw one that was up to 100 snakes per hectare.
That was the peak estimate exactly.
Right, but then, as you just made reference to, populations did go down somewhat.
Yes.
But not before they pretty much devastated.
Exactly.
Not just the birds.
Yeah, well, 10 out of 12 endemic bird species have been extinct. There are 2 that exist only in captivity. And then, they systematically, wiped away birds and then switched predation to rodents and other and native lizards on the island. Some invasive lizards as well, because there's other invasive species there. But have even adjusted to eating carrion, which is an interesting. So that's dead animals, which is an interesting thing for these snakes to do.
Very cool.
And there was even one native species of bat that has been severely reduced. So really, whatever they could find, that was appropriate prey. But just to add one more thing to what you said about the birds, one quote I found in a study was there was a near total loss of native forest birds. Because now when they're thinking about reintroduction, that's basically their starting point.
Yeah.
It's just this near total loss. And it's not just the birds and these vertebrate animals. It really affected ecosystems across the island, right? Because think about it, if you get rid of all those birds, all of the seed dispersal that they're doing is suddenly gone. So you have these species that offspring are basically stuck near the parent plant. And that's also going to greatly impact all the insect populations that the birds were feeding on.
Snakes are also eating all the insects.
But the one weird thing I saw was that spider populations, certain spider species, increased dramatically.
Interesting.
So just changes at many, many different levels because of this one species.
It's just that all that opened up real estate. Just the spiders were able to spread out more.
Right.
What preys on spiders? I actually don't know what's eating them. Are birds big spider eaters?
They must be.
Yeah.
Right?
Oh,
Yeah, that makes sense.
Yeah.
So that's where we're at with kind of what I wanted to share with kind of just give people a baseline of what happened on Guam. And then let's talk about what you're going to talk to us about today.
Awesome. Yeah, great introduction. There's those studies that show the sort of cascading effects of wiping out the bird population are pretty alarming, and that's why the brown tree snake is often used as sort of the textbook example of the negative consequences of I mean, quite literally the textbook example. I mean, you see it in a lot of textbooks.
Right.
Because of how widespread and how sort of all-encompassing the negative impact has been. And so the part of that we will talk about mostly is what our study focused on, Steve and I, as well as my former professor, Steve's current professor, Trevor Krabenoff, and our collaborators at the U.S. Geological Survey on Guam and actually the Hawaiian Islands. They're working closely for the reasons I mentioned before, focusing on the brown tree snake on Guam. The aspect that we'll focus on for the episode is what we focused on in our paper, which was the phenomenon you described where the snake was introduced to Guam with just a handful of individuals. Previous studies show it could be maybe as few as 10 individuals, that then later became this massive booming population, and a population that seemingly is able to adapt to its new environment, which will become the reason that's interesting will become apparent in just a few minutes. But what we were interested in is what happens to the genetic diversity of a population that goes through that type of what we call a bottleneck, where you go from this big, large population in their native range to just a handful of individuals in the introduced population, and then eventually, become this large, booming population on Guam.
Yeah, because typically, for someone like me who doesn't know a lot about genetics, if you have 10 individuals, you're going to have a lot of inbreeding. So shouldn't there be a lot of issues within that population genetically speaking?
Exactly. This highlights that highlights a really good point that we focus on in the paper, which is this genetic diversity population viability paradox, which is just this often observed paradox where individuals that have, like you said, populations that have very few individuals tend to be highly inbred, because there's a limited amount of genetic diversity if there's a limited number of individuals. So that translates rather quickly. And in the field of conservation genetics, there's this theory known as the extinction vortex, wherein when a population becomes very small, they become more inbred, and the more inbred they become, the less viable their offspring tend to be, and that leads to a further population decline. And this sort of extinction vortex is where they continually spiral into the point where they're extinct,
spiraling down the drain.
Yeah, and I think it's worth mentioning that when we talk about why this happens, is that let's just talk in the context of a bottleneck. So let's say you have this nice, healthy population to start with, and it has all this standing variation. And
let's start with Adam and Eve.
Yeah. So when we think of evolution isn't like, let's say if there's some kind of hurdle within this generation that a species has to overcome. It doesn't just, the variation to make it past that hurdle doesn't just come out of nowhere. It's something that already exists in the population. Yeah. So evolution works on standing variation. So if you have a lot of variation within the population, it's more likely that a species will evolve, or survive, right? Or a population will survive. Now, if you take that big population with all that variation and you limit it down to now 10 individuals and have them put in a new area where they're closed off from this old population, now you have a much smaller standing variation for evolution to work on and to overcome disease and
evolution
pests.
Has fewer options.
Yeah, so
yeah. Am I getting this right, Chris, that the brown tree snake should have, and I'm putting that in quotes, shouldn't have been able to explode into millions of individuals?
Yes.
Because with only 10 to start from, you wouldn't think they're going to make it.
Exactly right. Yeah, and that's what makes them invasive species and introduction events like this so interesting to study, because it's sort of like studying what would happen in the best case scenario for an endangered species that we're trying to recover. So the context you can sort of think of is, say we have there's an invasive rattlesnake, an eastern Massasauga rattlesnake that's currently on the endangered species list. And it's because of habitat destruction and stuff, it's gone down to a very small population size. And so it's interesting for conservation geneticists to understand how do populations overcome that type of impediment. And so by studying invasive species that in a sort of horrible natural experiment overcome these bottlenecks, by studying these populations, these types of examples like the brown tree snake, we can learn genetically and ecologically how do these things overcome such bottlenecks, which could actually potentially inform management decisions in the future for endangered species.
You're making lemonade out of lemons.
That's right.
Steve didn't like that one.
What a simple comparison.
No,
It's so simple it's lost any relation.
meaning.
And so to go back to the genetic diversity population viability paradox, the paradox, it's also known as the paradox of the invasive species, is that, like you said, the brown tree snake, if we were studying it as if it were an endangered species back in the 1950s when it was first introduced to Guam, we would say, oh, this thing's endangered. It doesn't have the genetic diversity required to evolve and adapt, and it looks like it was able to on Guam. And so it's fascinating for that reason.
But I also want to point out, because, sometimes when we talk about, and maybe we're opening a whole can of worms here, but I was just talking to Daniel, former co-host, about the ivory-billed woodpecker. Because he wants to go do an episode on the ivory-billed woodpecker down south. So one of the arguments that, at least in my head, with, again, the little that I know about genetics, when people talk about that, I always say, in order for there to be a viable population of those birds, you're going to need to have, at least several dozen individuals or more, right? And I wonder if some people will hear this and think, oh, the brown tree snake did it, so any species can do it.
Right.
But am I accurate in saying the brown tree snake, and one reason you're studying it, is it's an exception. Like, it shouldn't have happened this way. Is that accurate? Or?
Yes, it's accurate in one sense, but I think what is maybe a more important takeaway is that in this for this study, we show that the context really matters. Right. So the ecology of the animal really matters. So
and where it is, probably.
Exactly. Where it is, what it's surrounded by. So what you were describing earlier about all these naive prey, this is a phenomenon known as enemy release that you see with invasive species. And it extends beyond just what they have to eat, but what eats them and what infects them. So if you think about in their native population, in Papua New Guinea and northern Australia, there are many things that have evolved alongside brown tree snake that are have developed the ability to avoid them. So this is the naive prey argument, but also eat them. And the same thing is true for pathogens. So there's a lot of pathogens in their native range that would potentially wipe out some of these individuals that are inbred on Guam, that don't have the diversity required to overcome some of those impediments. And so when they're released from those things, when they're introduced to the new population, it's possible that the genetic diversity required to survive and overcome that sort of evolutionary arms race that it's in its native range is no longer imposed on the brown tree snake on Guam. And so it's sort of this, what we call like relaxed selection potentially, where in its native range it doesn't have to have, or in its introduced range it doesn't have to have as much of those, much of that diversity as it might need in its native range.
It's funny to think of it as prey, because we're thinking of it as this such destructive predator,
Yes, exactly.
But it's a prey release. So I'm going to propose this, and just don't say anything now. Let's have the audience really internalize this first. We should take the Massasauga rattlesnake and just start introducing it places and hope that it goes invasive, and then maybe we'll see some cool effect similar to the brown tree snake. I think it's a perfect strategy. But
what could go wrong?
I love it. But another important point of context, and this is really the focus of our study, is that it's not all genetic diversity is created equal, and we provide evidence that the ways in which we are measuring genetic diversity historically, through the mostly driven by technological limitations and sort of a hammer and nail problem with what we use for DNA sequencing. The way we were looking at genetic diversity is important too, and the types of genetic diversity and where in the genome those types of genetic diversity actually make a big difference. And so I think just to double back, I think the thing that our study shows well, and in fact the brown tree snake and other invasive species show well, is that it's not that genetic diversity isn't important, and it's not that small populations aren't endangered in their native ranges, or small populations are endangered when you're dealing with an invasive species. It's just that to get a more complete picture of how threatened or how at risk that population is in the context of its genetic diversity, you have to take a lot of these other aspects into account.
So you're saying ecology is complicated?
Absolutely. Absolutely, yeah.
We say that a lot on the podcast.
Yeah, we do.
It is, yeah. And a lot of genomic studies and a lot of genetic and evolutionary studies, in my personal opinion, sort of neglect to account for the ecology of organisms.
Well, as we often say, you don't know what you don't know, right?
Right, yeah.
So,
Well, just the perfect example, like we juxtapose the brown tree snake situation to the eastern Massasauga rattlesnake, which again is the endangered rattlesnake, to the brown tree snake in our paper, because we show that the level of genetic diversity using the traditional metrics by which people measure how genetically diverse and how genetically healthy. I'm putting that in air quotes for people who are just listening, which is everyone because there's no video for this.
You're going to get the ASMR of your gloves scraping in just the right way for people to know.
Yeah. So using the traditional metrics by which you measure the genetic health of a population, which is a very important metric because we use those types of measurements to determine what management actions we take for certain endangered species. But using those metrics, we show that the brown tree snake is currently just about as genetically depauperate or genetically inbred as the eastern Massasauga rattlesnake. But then you compare the eastern Massasauga rattlesnake is believed to exist in these small populations of 50 or fewer individuals to the brown tree snake, which is millions of individuals on the same island with seemingly very limited limitations to gene flow. So it shows that using these so-called what we call summary statistics, where the traditional metrics for measuring genetic diversity in a population, you would think that the brown tree snake is just as at risk of going through the extinction vortex as the eastern Massasauga rattlesnake, and obviously that's not the case.
So why isn't it?
Yeah. So yeah,
I know it's a simple question.
There must be something going on.
Yeah. And so one of the things that we highlight in the paper is that so I mentioned these summary statistics for genetic diversity. Two of them that I'll mention, and people can look them up, but there's an effective population size, which is essentially the number of individuals that contribute to a future generation in terms of reproductive viability, and heterozygosity, which is just the amount of genetic diversity at certain places across the genome. And we typically summarize these measures to represent. We sort of calculate these across the whole genome and then boil them down to one metric, and it represents the whole either population or individual. And
do you mind if I make sure that? Did you understand the effective population size?
Yeah.
You do?
I think so. Yeah.
Okay, I'll give just the most extreme example. Let's just say we have a population of 1 million individuals, and maybe we could also say like all their genes are identical to each other. This would so their population would be 1 million, but their effective population would be 1.
Yes.
Because there's only one copy of the same gene in every single individual.
Right.
So it gets kind of complicated because each gene can have its own effective population size, but we can kind of take like an average or.
And I was thinking as you were saying it, like that word effective. Mm-hmm. It's also very context dependent, right?
Yeah.
Not just within the genome, but like in the environment that you place the animals into.
Absolutely.
Right.
Mm-hmm.
So yeah. So you were saying they boiled it down to one metric.
Yeah. So you have these sort of summary statistics. And that's a great clarification, Steve, because it might sound I might have sounded crazy saying that the effective population size of population of snakes on Guam, which is 2 million, was nearly identical to that of a population of 50 eastern Massasauga rattlesnakes. But it's because of this inbreeding characteristic. Because in many parts of the genome, they have these things called runs of homozygosity, which is two copies of the genome. So snakes are diploids like humans are, which is means they have a copy of their genome from each of their parents, and so they have 2 copies of every one of their chromosomes in every one of their cells, and one of those copies came from their mom and one of them came from their papa snake.
You can kind of think of it. I had a professor that used to say that everyone is a population of 2.
Yes.
Because we have 2 copies of our genome inside every single one of our nuclei of every single one of our cells. So we have 2 copies of our genome in every single cell. So we're a population of 2, kind of.
Nice.
But, yeah.
Yeah, and so one really good way of measuring how inbred a population is to look for stretches of the genome within an individual that are identical by descent, meaning they inherited identical copies from both of their parents. And you can imagine how this correlates with inbreeding, because if full siblings breed, then about 50% of their genome or more is going to be identical by descent because they inherited the same copy from their parents.
Kind of like Bill's genome.
I was waiting for you to say it.
I'm sure I have some long runs of homozygosity coming from eastern Kentucky.
You can fix it in one generation, though.
I don't need your woke diversity in my body.
All it takes is a generation to fix inbreeding, though, right? Isn't that true?
Potentially, yeah.
Yeah.
Absolutely.
All right, so Violet's okay.
Depending on, yeah. Yeah, there you go. And so when we look at this, when we identify these across the genome and then summarize the proportion of the genome that's comprised of these runs of homozygosity, we call it FRO, which is just fraction of the genome in runs of homozygosity. That statistic is very similar in the eastern Massasauga rattlesnake and the brown tree snake. But what we did to provide further context to the genomic consequences of the bottleneck that the brown tree snake went through was two things. One, we instead of looking at these genome-wide summary statistics, we looked specifically across the genome where in the genome do these inbreeding events occur, and where, if anywhere, is there actually remnant genetic diversity. And we were able to do this because I was able to produce a really nice high-quality genome assembly, which you can just sort of think of as a really nice blueprint for where things are in the genome. It resolves genomic architecture, meaning what genes are in the genome, where they're at relative to each other, where they're at relative to things that affect how genes are used.
Do you mind if I just give a quick example? Because both
Please.
of us have worked with really, really, really poor genome assemblies, and I'll try to explain it as simply as I can.
Yes,
so
please.
just using the human example, everyone's aware of 23andMe, that website that does people's genomics.
That constantly leaks your data to third parties? Yeah, go on.
Yeah.
Heard of
Well,
them?
so if we were going to do a genome assembly of the human genome, we would have, because it's called 23andMe because there's 23 pairs of chromosomes in the human genome. So if we were going to do a perfect genome assembly of the human genome, we would have 23 strings of A's, T's, C's, and G's that would make up the entire genome. Well, actually, we would have 23 pairs of A's, T's, C's, and G's that would make up the entire genome. Now, I've worked with some species that, because of the sequencing technology, you go to do the genome assembly, and you're looking at it, and instead of 23 pairs of sequences that are very long, it's like 100,000 tiny little pieces of sand. Instead of 23 big chunks, it's like you're looking at a line of sand instead. So you can imagine that when you have these 23 big chunks of DNA, you can see like, where is gene A relative to gene B? Where is gene B, relative to gene C? Is gene A through F in a different order on this chromosome pair, or you can actually figure out this interesting positional data if you have the whole chromosome. But when you're dealing with these like really poor chromosomes, these like poor assemblies where it just looks like sand, you can't get any of that positional data. So I've worked with both, and I will say when you have the whole chromosome assembled, from telomere to telomere, all 23 pairs of chromosomes, it's so much more interesting.
What's telomere?
Those are just the ends of the chromosomes. There's these little repeat segments.
So, Chris, you said you were able to create a really good assembly for the brown tree snake?
Yeah, so Chris isn't looking at little pieces of sand. He's looking at full chromosomes, and actually not just full chromosomes, but he's getting the pairs of chromosomes.
Yeah, it's actually,
well.
we were able to phase the genome, meaning that we were able to not only assemble all 18 chromosomes in the brown tree snake karyotype. The karyotype is just how many chromosomes you have in your genome. And we were able to assemble the maternal and paternal haplotypes separately. So we were able to assemble the copy that this individual got from its maternal line and the copy of the genome it got from its paternal line separately, which provides some really cool context for specific evolutionary consequences, including sex chromosome stuff, which maybe we'll get into in a bit. But yeah, as I was saying, by having this really high-resolution version of the genome, we could identify not just how much genetic diversity is there, but specifically does it cluster in one region or another? And if it clusters in one region or another, are there any specific genes that it clusters around? And that can provide important functional insights into how the genetic diversity might be actually affecting the fitness of that population. But another thing that we looked at, in addition to the sort of locus-specific regions where genetic diversity might still be harbored in this population, we looked at these types of mutations, a different type of mutation called a genomic structural variation, which Steve mentioned at the top of the podcast. At the top of the episode. So all the genetic diversity metrics I've been mentioning previously in this episode have relied on what we call single nucleotide polymorphisms. These are mutations that affect just one A, T, C, or G in the genome, and there's been a sort of hyper focus on these types of mutations because of the technology that we use. So without getting too much into the history of sequence technology, the methodology for sequencing the genome that is most popular, or is at least most popular for the last decade, is a process wherein you take the genome of an individual, break it up into pieces that are about 150 nucleotides long, which is quite small in retrospect, and then you sequence those little 150 base pair pieces, and then if you think about trying to do downstream analyses with individuals, let's say just a human, for example, which has a genome of around 3 billion base pairs, 3 billion nucleotides long. When you try to use those 150 base pair sort of puzzle pieces to figure out what's going on in a 3 billion base pair genome, comprised only of 4 nucleotides in total, it can be quite difficult to reassign the bits that you sequenced back to their proper location in the genome.
You can kind of think of it as like, just to make it very simple again, but you're putting together a puzzle.
Yeah.
And with this older sequencing technology that is still super useful, and we actually still use it today, and it's amazing. But the technology he was talking about, this is where these little grain of sand genome assemblies, you can make really, really nice genome assemblies with this old technology. But I've also had the case where you make these grain of sand genome assemblies, and they're kind of rough to work with. But the analogy I want to use is putting together. Oh, and I'm sort of jumping ahead, but contrast this against these long read sequencing where you can get reads as long as like 1 million bases instead of just 150. It's like putting together a puzzle with like tiny, tiny little puzzle pieces, and a lot of those puzzle pieces actually look the same. I'll just sort of add that in there. Versus putting together a puzzle where the puzzle pieces are like massive, and it's almost like a little kid's puzzle piece, or at least something closer to a little kid's puzzle.
Yeah, which is much more my level.
Yeah, with young kids, Chris and I get a lot of practice with these little, these simple 2-year-old puzzles.
Yeah, and so as Steve mentioned, this technology we call long read sequencing technology. You're able to sequence the genome. You only have to break the genome of individuals up to tens of thousands of nucleotides long, up to millions of nucleotides in length. And so with these much larger puzzle pieces, you're able to resolve the structural architecture of individuals' genomes much more readily. And while this has led to extreme advances in genome assembly for the puzzle piece reason that Steve just mentioned, and that is indeed what we use to assemble the genome of the brown tree snake, we also generated this type of data for multiple individuals from the brown tree snake population. And what this allowed us to do was to use these really long puzzle pieces to look for mutations that affect more than 50 base pairs or more. And these are known as structural variations, which I mentioned before. And if you think about the way that type of mutation is categorized, 50 base pairs or more, you can imagine how difficult it can be if the mutation is 1,000 base pairs long, how difficult it is to use this old sequencing technology of 150 base pairs in length to try to characterize some of these things.
Yeah, just to give some real-world numbers, so when you do the old type of sequencing, your average piece is 150 base pairs. The sequencing Chris and I have done, like I've done genome sequencing where half of all my sequencing is like 30,000 base pairs. Like each piece is 30,000 base pairs, not 150, 30,000 base pairs or bigger. Half of all my sequencing. So you can imagine the advantage to instead of getting little tiny.
Exactly. Yeah.
So if you're looking for things that are 50 base pairs or longer, I mean, I don't know.
Yeah.
Or longer is the big piece. Because what's the one we just found in the study sample we're working on right now?
900,000 base pairs, yeah. And there's another one
Crazy.
in my char population that I found that's also over 900,000. Again, something that would be nearly impossible to identify with this previous technology. But by generating this data for multiple individuals within the Guam population, we generated what is now referred to as population scale long-read sequencing. And so we were able to identify these types of mutations in multiple individuals from Guam. And so using those data, those long-read data, we looked for these structural variations in multiple individuals and then asked a similar question. What type of diversity is there in those types of mutations, and what types of genes are they affecting? Where in the genome do they occur? And if they occur in one spot more frequently than in another, what are the genes in that part of the genome? And using this combination of what I mentioned, what seems like an hour ago, the locus-specific investigation of where these single base pair mutations are, so like where in the genome and what they're affecting, and looking at these structural variations, we found that although the population of brown tree snake on Guam is in fact inbred using these summary statistics that we mentioned, they do harbor some interesting diversity both in these single base pair mutations and in these genomic structural variations. Importantly, the latter of which would have been missed if we only used short read sequencing, or if we didn't look at this locus specific approach.
So even though you didn't have, see if I'm understanding what you're saying, even though you didn't have a ton of individuals, possibly there was enough genetic diversity within those individuals.
Yes.
To allow this situation to happen.
Exactly. And so what I think it highlights is, yes, genetic diversity is important, but the type and where genetic diversity, the type of genetic diversity there is and where it occurs is also very important.
So if you're taking like a genome average, that might not actually give you the proper insights into its like survivability or not all genetic diversity is created equal. It
Right.
feels like so.
Exactly.
Sometimes it'll be in the right spot for the right scenario.
Now,
Yeah.
can I ask you a question?
Mhm.
And we can cut this out if,
Yeah.
because I'm just, I'm thinking as you're talking.
I'm sitting on
No, the audience might be asking the same question.
Yeah,
So
probably.
So if say they started from 10 individual brown tree snakes on Guam, if there were a different 10 individual brown tree snakes, could the situation not have happened? Was that genetic diversity because of the individuals, or is that diversity probably present throughout the species?
So
Or-
I think it's a fun thing to think about, and I think about it pretty often. I think the safest bet would be to say that the diversity that we characterize in the Guam population is very likely a subset of what actually exists in the larger population in its native range. And for your first question, if a different 10 individuals were introduced, is it possible that they would have gone through sort of an extinction vortex? I think the answer is possibly yes.
Okay.
I do think that there's a certain amount of luck involved in these things. So if you think about how many, if you are keeping tabs on how many invasive species there are around the world, it's a daunting number. But I have to imagine that there are many more introduction events that didn't lead to successful establishment.
Right.
Yeah.
And it could be partially because of this thing, but probably also a little bit more random luck of, 10 snakes got introduced and they all 10 of them got run over by half of them got run over by a car or something like that. But
I think what Bill's saying is, let's say they didn't get run over by a car.
Oh, of course, yeah.
There's something different about the variation that they provided to this invasion zone
Exactly.
that didn't allow for this, this variation in the right places, or
But it also doesn't exist in a vacuum either, because it's the conditions that are present on the island too.
Right. The perfect invaders could always come into a place and then get run over by a car.
They get run over by Steve coming late to the podcast.
Yeah. Oh, well, we don't talk about that. Actually, we mentioned it on a lot of podcasts.
But also, just the why the argument that I made at the top, that ecology matters, right? So again, it's possible, and in fact, quite likely, that when the population on Guam went through this bottleneck, it was left with a higher proportion of what we call homozygous recessive deleterious mutations, which is a long way of saying...
Great band name.
Yeah, which is a long way of saying mutations that really only hurt the organism if they have 2 copies of that bad mutation. And these homozygous mutations, again, exist in the large population, but they're usually at small enough numbers, or the genetic diversity in total in the population is large enough that the likelihood of any one individual getting 2 bad copies is quite low. So it doesn't really harm the population. But when you drop down to 10 individuals, let's say just a handful of them have that, the probability that their offspring will inherit 2 copies of that bad mutation are much higher.
Way higher, yeah.
Yeah, much higher. And there's several cool arguments against how invasive species overcome that. One is this phenomenon known as purging, where it's so deleterious that the individuals just die, and then those types of deleterious homozygous mutations are wiped out from the population. In fact, there's an argument that they can actually be wiped out from small populations, bottlenecked populations, more efficiently than they can be in large populations.
Oh,
where
makes
they can
sense.
sort of exist in sort of background numbers for longer durations.
Mm-hmm.
Again, using the sort of the summary statistics, we saw that the brown tree snake was as inbred as the eastern Massasauga rattlesnake. But when we looked at the types of genetic diversity and where they were, one of the really fascinating things we found was looking at these single base pair mutations. We found that genetic diversity was clustered around immune function genes, and this is a common phenomenon that's been found in invasive species, invasive populations. And that is the sort of counter-argument to the enemy release phenomenon I described, where they co-evolved with a bunch of pathogens. A species co-evolved with a bunch of pathogens in their native range. They themselves have actually evolved. It's a co-evolutionary arms race, so they themselves have evolved the ability to fend off those pathogens. But when you're introduced to a new population, you're actually introduced to a bunch of pathogens that maybe you don't have a proper defense for. And so it's sort of a double-edged sword in that regard. And one of the things that we see in the brown tree snake population is that either genetic diversity was disproportionately maintained in these immune function regions, specifically this major histocompatibility region, which is a really important part of our vertebrates' immune systems, or it was potentially accumulated novel mutations once it was introduced to Guam. And so regardless of whether it was sort of a carryover, whether they maintained the diversity that they brought with them essentially, or they accumulated new mutations there, we found genetic diversity was really clustered in these major histocompatibility regions, which again provides this sort of nuance to, if we just looked at these summary statistics, we would be potentially missing the type of genetic diversity and where in the genome the genetic diversity. So it's sort of argument to how do we get to the functional importance of genetic diversity. So like if Steve and I were to compare our genomes, we would find approximately, I think, 3 million differences. But a large majority of those differences between our genomes would occur in what we call non-coding regions, or intergenic regions where there aren't any genes and very likely have little to do with how genes are expressed. And so if we then boil that down to mutations that actually affect how genes are used, we would have a much better picture of how Steve and my genetic diversity, the functional diversity differences between our genomes actually occur. Yeah, so that the major histocompatibility finding and the immune gene finding highlights the importance of not just summarizing genetic diversity across the genome, but looking at where it's at. And in terms of the structural variation finding, what we discovered by using an approach that allowed us to characterize both the small mutations and these larger structural variations was there was a large number of structural variations, 19,000 structural variations, that were clustered around genes that affect olfaction, so how snakes smell. Specifically, there were a lot of mutations in these genes called V2R genes, which is vomeral nasal 2 receptor 2 genes, and these are the primary olfactory receptors in snakes, and there are a bunch of copies of these genes in snakes because they've evolved a propensity for relying more on olfaction than other animals. And we found a lot of genetic diversity in the form of structural variations affecting these V2R genes. And what's really fascinating is in the literature, in snakes, in humans, and in mice, these V2R genes are responsible for smelling genetic diversity in their mates.
What?
Specifically in the major histocompatibility complex genes. And so, it's really remarkable. So in humans, they've done these studies where they test partners or mates, and the probability that your mate has a different version of the major histocompatibility genes than you is much higher than you would expect by chance.
I've heard about that.
And so there's actually evidence that in humans and mice and other animals, they select for different major genetic diversity in immune function, which there's a strong evolutionary argument for why that's important. But sort of the added layer of coolness is that these V2R genes, specifically in mice, there's some studies you can't do in humans that you can do in mice because of how you have to treat the study animal. But they've shown that it's these vulnerable nasal gene receptors that really are able to detect this diversity. And so the sort of picture, and we haven't tested this, future work will need to be done to validate this, but it seems like maybe the mechanism for maintained genetic diversity in the brown tree snake, for where it exists in their genome, is maintenance of structural differences, genomic structural variations in these vomeronasal nasal genes that that maintain diversity in how these animals can smell and detect mates, specifically to detect variation that's maintained in these major histocompatibility genes, this immune function genes. And so, again, it's just getting more towards the nuance of how is the genetic diversity maintained and sort of where it's maintained. And this is potentially a sort of cool story for, you need enough diversity in your vomeronasal nasal receptors to sense diversity in major histocompatibility, and you need enough diversity in your major histocompatibility genes to have enough diversity to be detected by the vomeronasal nasal receptors, and you need the diversity to battle novel pathogens. And so it's just a really neat story that's sort of all hinting towards this sort of mechanism.
It's wild. I'm just imagining a snake going up to another snake and just, girl, do you got that 50 kb insertion in there?
So there's these kind of two areas you discovered that was an possible advantage for this population.
Yes.
Small population that allowed them to balloon into this massive overtaking of the island.
Yep.
So
it just dawned on me that I said a 50 kb insertion while making a sexual joke.
Yeah.
I actually didn't do that on purpose. I was just, it was a DNA insertion. That's a type of SV.
I didn't even think of.
Did we say what type of? I mean, what are examples of SVs?
No.
Yeah, we should say, because I think the major thing we said about SVs is just something in the genome that affects something that's
structural variants.
Structural variants are things in the genome, or affect something in the genome 50 base pairs or larger.
Right.
But we didn't give examples of what that could be in the genome.
That's true. Yeah, so there's 5 flavors of structural variations, and one is a deletion, and this is just where exactly what it sounds like. A segment of the genome is deleted, so it's absent from one individual that might be found in another. Another is an insertion, which is the exact opposite, where a segment of DNA is inserted into the genome, into a specific location of the genome that isn't there in other individuals. A duplication, which is just a part of the genome that's duplicated. It can either be duplicated right next to the original copy of that part that's duplicated, which we call a tandem duplication, or it can just be a duplication from a different part of the genome into another. And then there's translocations, which is where a chunk of the genome moves where it's located relative. So it could jump to a different chromosome, or it could jump to a different part on the same chromosome. And then there are chromosomal inversions, which is, if you think about the genome as an individual chromosome as sort of like a long ribbon, if you took a segment of that ribbon and flipped its orientation, but it stayed on the same chromosome, that's called a chromosomal inversion. And another thing I should mention about structural variations that are really interesting and important, and something that's come to light after the advent of this long-read sequencing, is that because structural variations are bigger, they're 50 base pairs or more, whereas single nucleotide polymorphisms are a single mutation. On average, or per mutation, structural variations are much more likely to actually affect how the genome functions. And so if you're affecting a bigger chunk of the genome, you're more likely to affect either how the gene functions, or whether the gene functions at all or not, or these regions that regulate how genes are used. And so structural variations are more often associated with actual phenotypic consequences. So how the organism moves, thinks, feels, reproduces, all of these things.
So even if you have, let's say, you have two individuals and they have all the same genes, the only difference is the gene order or something's different about where it's positioned, that can change the activity of the gene or how often it's expressed, and so you can get very different things in those individuals just by having those genes in some kind of different orientation or copy number.
If you have two copies of the same gene, you get more of the product of that gene oftentimes, and that can affect the stoichiometry or sort of the balance of gene products, which can all have important consequences on the fitness of an animal or plant.
All right, so now it's probably a good time to shift to talking about management of the snake as it is now, because we alluded to before that after the population spiking in the 80s, populations settled, but they're still rampant across the island. And we haven't really talked about management efforts. And we should also point out that it hasn't just impacted the wildlife, it's also impacted people. We mentioned they're mildly venomous. So when people are bitten, most people recover, but it can cause problems with the young or infants, and they do get into houses, which is kind of terrifying.
Yes.
A 2-meter snake coming into your house.
Hearing about how they get into houses made me think of how they probably got onto ships or whatever else.
Yeah.
It's like there's just sneaky little
And then
snakes.
how did they cause power outages?
So they wrap themselves around power lines and will connect two power lines that are meant to be separated, or they'll wrap around like I think transistors or something. I'm not an electrician, but they reportedly cause around $2 million of economic damage every year from causing these power outages.
Like, whoa, they built me a tree. Sweet.
And then they're also eating livestock.
What?
Pets, things like that. So
livestock? What am I thinking? Oh,
well,
like chickens and stuff. Okay.
So,
sometimes I forget that they're livestock too.
And from
mostly
what I
think
read,
cows and stuff.
the human impacts is really what's driving more of the funding of trying to control these snakes.
Oh yeah.
The human impacts.
Yeah, there's a military base, as I mentioned, on Guam, and they fund a lot of research because they're impacted by the power outages and also probably get bit by snakes every now and then.
I'm just imagining a Starship Troopers like scene of like the military base just like defending against the snakes, the 100 per hectare, just racing towards
a
them.
wall of snakes. So a lot of the management efforts I've read about are pretty traditional. They're either capturing snakes with traps, they're putting out baited prey, trying to poison them, and pretty much everything that's been done so far has been, it can be effective locally.
Yeah.
And if you do it constantly.
Yes.
But they would have to like if you want to reintroduce some species that have been lost, the statistics I saw, you would have to eradicate like 80 to 90% of the snakes that are there and keep them at bay.
Exactly.
They get rid of all but 10.
Right.
Oh no.
So your research, though, has opened up kind of this whole window that I had no idea about. So tell us about that.
Yeah, so there's the possibility for using genetic controls for invasive species, and this is not my area of expertise, and I am also I think we need to do quite a bit more research before we could ever confidently implement some of these things. But by knowing the type of genetic diversity and where it occurs in the genome and the functional aspect of that genetic diversity, managers could potentially develop a strategy that leverages that information, sort of identifies a weakness in their genome, for example, or if they wanted, this is an extreme example that I hope never is used, but let's say they wanted to engineer sort of a virus that only targets the brown tree snake. If you were ever to get to that point where we could do that safely and effectively, this genetic diversity information we have, and specifically the really high quality genome assembly we have, would be essential for that. So you could find a region of the genome that, let's say, is identical in all the snakes that we've tested. That represents a real vulnerability if we were able to target somehow that part of the genome that they all have identical copies of. And so that type of information, that's sort of an extreme sort of dystopian example, but...
so you're not advocating for that?
No.
Wait, we live in a current dystopia because I heard that these snakes are also vulnerable to autism. Because
right.
Aren't we feeding them acetaminophen?
Yeah, that's right, yes. Yeah, the aspirin baited traps, acetaminophen baited traps is an effective... Well, it's not effective because it doesn't seem to be eradicating them, but it does kill the snakes when they eat them. But
then they get autism.
I'm just
in the afterlife.
I'm just sort of spitballing this here, but something to think about is we found this diversity in olfactory genes, right? And the way that these animals find the bait in these baited traps is through olfaction, and so it's possible that this could somehow inform how they're baiting their traps, or if they need to increase the diversity of scents in each trap, or a fun theory that one of my co-authors on this paper, Levi Gray, pointed out was that the brown tree snake on Guam don't exhibit cannibalism, whereas in their native range they exhibit a ton of cannibalism, or a considerable amount. And so one idea is that through these olfaction genes, maybe they're able to say there's an evolutionary propensity to not eat your close siblings. In their native population, where the population is much larger and there's much more diversity, any individual brown tree snake that another brown tree snake encounters is much less likely to be a full sibling, or a close enough sibling to deter it from being eaten. But on Guam, where they're all so highly inbred, maybe they're all smelling each other and thinking, oh, you're a close enough relative based on whatever genetic diversity may be in their major histocompatibility region, that they don't eat each other. And it's just this sort of interesting consequence of this bottleneck that they went through. And
I guess they're all siblings. They're all kind of siblings in a way.
Exactly.
Cousins.
Yeah, exactly. And so, that's one aspect. So just knowing how this diversity works, how this diversity exists in their genome, you could use something like their olfaction to potentially target. But I think a lot more functional type studies would need to be done to really inform that type of stuff. And there's some really cool work being done now to look into those types of things. But an important... I mentioned earlier how studying invasive species can help inform how we handle and how we manage species that are endangered or species that are at risk. And so one of the interesting things that we found, or one of the interesting takeaways that I hope we can sort of get from this study, is that when we're assessing a population that we consider to be endangered or threatened, when we're assessing their genetic health, hopefully we can take these more nuanced approaches to determine whether or not that population is imperiled as a result of them being genetically depaupered or them not having enough genetic diversity. Because one of the current approaches to overcoming that barrier, when, let's say we find a population, let's use the eastern Massasauga rattlesnake for convenience. Let's say somebody decides that they're too inbred to survive on their own, they're going to go through the extinction vortex. What can we do to manage that? One thing that they're doing is a notion or a phenomenon, or is an approach called genetic rescue, and that's where they take individuals from one population and introduce them to another population. Say these two populations would never have a chance to reproduce in the wild, but they have their own types of genetic diversity. If you take individuals from an outside population and bring them to a new population, you're introducing novel genetic variation, and this is perceived to be an overall good thing because you're increasing genetic diversity in that population. But what can happen, and what is sort of a reason that we should be hesitant to do this until we're really sure that the population actually needs some sort of rescue, is this phenomenon known as, or it breaks up this thing called local adaptation. So let's say, for example, that the population that you decide is genetically in need of rescue has some adaptation that makes them really well fitted to their current environment. If you bring individuals from outside that population that don't have that didn't evolve in those same conditions, and you introduce them to this population that you're trying to quote unquote rescue, you could actually break up the genetic underpinning that makes that population that you're trying to rescue so well adapted to its environment, and it sort of breaks up, like I said, this phenomenon known as local adaptation. I think that with the findings that we came up with in this study, it will hopefully encourage people that are conducting conservation genomic studies and assessing the health of genetic health of a population. Hopefully it will encourage them to look at these more nuanced, look at the genetic diversity in a more nuanced way in the populations before resorting to genetic rescue.
And longtime listeners, they might see a connection here because we did an episode on the Florida panther.
Yeah.
And that's what was done was mountain lions from another part of the country were brought in, but there was a long debate about that.
Yeah.
Whether to introduce, because it's a subspecies, so they are going to have some different genetics. So that could have gone badly.
Yeah.
Potentially.
I'd say
Luckily
this also
it did not. There's a recent study in the Proceedings of the National Academy of Sciences that showed that it worked out good. But it didn't necessarily. I mean, it's not guaranteed that it would have happened that way.
Right.
And I also think that study, the study that said that the introduction, the genetic rescue was successful, failed to look at genomic structural variations and didn't focus enough on the types of genetic diversity. So it's possible that there was some sort of local adaptation in the Florida panther that was lost or broken up, but they may not have been looking at the right types of mutations or in the right parts of the genome to really have detected that.
Do you think, and I know this is a big ask, do you think consequences of that action in the Florida panther could still play out in the future where we could see a sudden loss in panthers in southern Florida because of some genetic adaptation that we've wiped out by introducing?
Absolutely, yeah. I think that's perfectly plausible. Even if we didn't fully wipe it out, if we knocked it down to a low enough frequency in the population that it's either rare or unlikely to rebound to a high enough frequency to really make a difference in the population. But yeah, it's perfectly possible.
Yeah. So it's no matter what choice, it's risky, right?
Yeah, exactly. And again, if there are it's not that we're against these types of management actions. Sure. Endangered species need our help. I just think that, and there's a good body of literature that support this, 9 times out of 10, the best thing you can do is actually to reestablish or remediate their is to repair their habitat. I mean, habitat remediation is by far the most effective means of...
Which we just removed from the Endangered Species Act.
Yeah, right.
Oh, yeah.
Shocking.
Uh-huh. Right?
So we increased the Florida panthers to a population of 1 million.
Yeah.
But they have nowhere to... But somehow they got that high,
Wow.
It did remind me, we do have a local ecotype episode that reminded me kind of the importance of the genetic information from individual area or specific areas and how important that is. And then also just personally, I was in a program called the Seeds for Success program, and you were not though, where we were doing seed collections from very specific spots that we took the GPS coordinates from it was like 12 states out west, and those seeds were actually collected for future reseeding efforts. So if that land had something had to happen to it, and they wanted to reseed the land with the local genetics, local populations, they could do it because we did those seed collections.
Yeah, and they'd have probably a better chance of success than ones from the next state over.
Right.
Yeah, very cool. All right, so I think a kind of a good wrap-up statement is a lot of the things that you've talked about, I like how you couch them in, these are potentials. And this research that you're talking about opens up all of these areas, but so often when you're dealing with endangered species, invasive species, the decisions you have to make are kind of which is the best, worst decision?
Right,
you know what
exactly.
I mean? They all have downsides.
Yeah.
So.
And Chris is really just saying to do a little extra work and look at those SVs,
No, an important point is that it's not some grand revelation we had necessarily, and it's not to cast aspersions at people doing conservation genomics. It's really just an advancement in technology and a decline in the cost of this type of technology that really facilitated this. It's not that nobody had thought about this before, nobody was curious about whether or not these types of things, it's just how much less expensive it is and how much more feasible. The sequencing technology and the sort of wet lab aspect of it, the chemicals, all of that has gotten so much cheaper to do these long read sequencing, but also bioinformatically, the algorithms and the computer programs that we use to detect these types of things have improved drastically. And so now you can do this type of study on a much more modest budget, which sadly conservation genomics projects often have more modest budgets than crop genomics projects do or livestock genomics projects do. And so it's becoming much more approach. I mean, I did 90% of the analyses from this project on a computer at our school, and we sequenced the stuff in-house. It's much more feasible from a conservation genomics budget standpoint.
You know, though, I think it's important to note that, like, this is the situation, like, all scientists are in. Of course, it's going to differ depending on their funding, but I like the saying that fortune favors the prepared mind. And so it's like, sure, all this technology is being advanced by other people, and these algorithms are being built by other people, but, like, you know, we play an important role as scientists, taking advantage of all this cool stuff now.
Course,
so,
yeah.
But the technology is going to keep getting better.
Yeah, and less expensive.
And I hit a ceiling, actually. It's not getting better.
Well, not for the next few years.
Yeah.
Oh, God.
Not without any money.
Yeah.
All right. Well, Chris, thank you so much. I feel like the second run through, I was able to stay with you guys, at least, you know, hanging on by my fingernails instead of just being lost for long chunks.
Yeah, thanks, guys. I appreciate the opportunity to be on the podcast, like I said, and thanks for keeping me on path when I'm a little brain fried here. So.
Thanks for powering through.
Yeah.
All right, first and foremost, we'd like to thank our growing list of Patreon supporters, and we also like to give a special shout out each month to our top patrons. So stick around for the end of the episode to hear my daughter Violet share that list.
And if you'd like to support the podcast, consider becoming a patron of our show at patreon.com. Because of support from our listeners, we've been able to keep the show free and make cooler episodes like this one. Or you can make a one-time donation through PayPal on our website.
Remember, you can also help out by sharing the podcast with friends and family, and by subscribing and leaving us a review wherever you get your podcast. It helps spread the word and allows us to reach a wider audience. We'd like to thank our newest iTunes reviewer, Sloth Travel Club.
Cool name.
Yeah.
So come enjoy our sporadic posts on Facebook, Blue Sky, and Instagram. And you can always email us ideas for episode topics, criticisms, or your own stories of personal encounters with genetics to thefieldguys@gmail.com.
And remember, folks, get those kids outside, let them get muddy, let them get dirty, let them flip over rocks and logs. And if you don't have kids, please make some time to get yourself outside. And folks, we'll see you next time.
See you next time.
And here's Violet with this episode's top patrons.
Eric, Alyssa, Adam, Colin G, Mary B, Renz, Abby, Dr. Judy, Kimberly B, Peter R, Callie, the Hebranks, Rich K, Rachel L, the drunk phytologist, Orange Julianne, DJ, Diane, we named the dog Indy, Hot Dog Cold Weather, Dwayne H, Jennifer D, Jonathan A, Deborah B, are the plants in the room with us now, Jeremy M, Lori B, Licky Weeks, Virginia A, Catherine W, Holly M, Michaela, Val, Andy W, John R, Evan, Picasso 3, Mark T, Cecilia, Julie B, Dennis, Chris N, Sung, Jeff S, Jonathan K, Matt E, Plants in My Pants, Ryan R, Sophie S, Connor H, Measure and Principle, Fragaria pepiloinoidea, Outside Chronicles, Quixote, Max D, Melissa Marie in Dusty, Arizona, Kelly S, Sarah S, Helen A, Judy F, Ben C, Jane H, Doodle Dude 82, Galen Mac, Kazis, Esther C, John W, Mark V, Bethany H, Rob M. Whew!
Well done, Violet.
Hey, that's a lot of patrons.
And patrons, on behalf of Steve and myself, as always, thank you so much for your support. It keeps us going. And we'll see you next time.
Bye.